Hello, grade 11 student. In this video, we're going to talk about our chapter genetics. So, chapter 2, activity 1, talking about the hereditary traits and the genes of 40 on the handout that you have already. So, chromosomes at metaphase, they are made up of two chromatids. The substance that makes up the chromosome is called chromatin. Chromatin is composed either of DNA that composed of a nucleotide ATCG or of proteins, histones, and non-histones. DNA, by the process of transcription, lead to the production of mRNA, and by the process of translation, mRNA is translated into polypeptide. Gene. What is a gene? A gene is a fragment of DNA that codes for a polypeptide. Alleles, different versions of the same gene. For example, blood group gene had three alleles, A, B, and O. A and B, they are dominant, and O is recessive. Homozygous or pure. Homozygous or pure individual that has two identical alleles for the same gene. For example, blood group A, the person is homozygous, blood group A and he's homozygous, so it have blood group A and have two alleles AA. Homozygous. Heterozygous hybrid is an individual that has two different alleles of the same gene. For example, AO, BO, or AB. This is the meaning of heterozygous or hybrid. And homozygous, same two identical alleles for the same gene, and it's called homozygous or it's called pure. Dominant allele, the allele that expresses itself in the case of both homozygous and heterozygous. Recessive allele that can be expressed except in the case of homozygous. Co-dominant alleles, alleles that are both expressed in heterozygous individual and a homozygous individual. This is the case of dominant. Co-dominant is different. It's expressed in heterozygous. For example, if we have a person that has blood group AB, so AB is co-dominant. This is the meaning of co-dominant. Okay, guys? Phenotype, the sum of traits found in an individual. Genotype, the sum of alleles, the genetic makeup of individual. For example, genotype, let's say for the blood group, it's AO. So A dominate O. So the phenotype is going to have blood group A. So always the genotype codes for the phenotype. True breeding line is a line of individuals where the descendants always have the same phenotype as the parents. Homogametic is an individual that gives only one type of gametes. Example, female ovum. You only give one type of gamete, it has XX, so it only gives X. Heterogametic, individual that gives more than one type of gamete. Example about this male, it has XY, so it's two different types. Ligous type, species whose males are heterogametic have X and Y. Example about this, humans. While Apraxis type, species whose males are homogametic have ZZ, and females they have ZW. Different sex chromosomes. Example about this, chicken, butterflies, and others. Hollandric gene, the gene that's carried by the Y chromosome, so it's found only in male. Bidominant, an individual having two dominant phenotypes. For example, blood group A and normal regarding the albinism. Both of them, they are dominant. So this is called bidominant. Birecessive, the opposite of bidominant, individual having two recessive phenotype. For example, blood group O and albino. Let's say small d or A small. Okay? Dihomozygous individual, homozygous for both traits. For example, have blood group AA, homozygous or pure, and for normal regarding albino having DD, so homozygous for both traits. Diheterozygous, heterozygous for both traits. For the blood group, maybe it has AO and DA for albinism. So this is called diheterozygous. In this experiment found in the book, we have a true breeding female black mouse was crossed with a male of the same genotype. And we have the nucleus of the embryonic cell is extracted and then injected into the fertilized ovum that is taken from a true breeding gray female mouse crossed with a male of the same genotype. So then the two pronuclei, they are extracted, and the obtained ovum is put in culture for 4 days. Then the obtained embryo is implanted in the uterine cavity of the white mouse, not fertilized, it's ready for gestation. So describe from a true breeding black female mouse crossed with a male of the same genotype, an embryo 16 cells is taken. The nucleus of one of the embryonic cells is extracted and injected into a fertilized ovum that is taken from a true breeding gray female mouse crossed with a male of the same genotype. The two pronuclei are extracted. The obtained ovum is put in vitro culture for 4 days. The obtained embryo is implanted in the uterine cavity of a true breeding female white mouse, not fertilized, ready for gestation. The result was the birth of a black baby mouse. So, analysis. After implanting the embryo obtained from injection of the nucleus, from where? From a black mouse, and injecting in the ovum of a pure gray mouse, and injecting the uterus of a white mouse. So we take the nucleus from the black, the ovum from the gray, injecting into the ovum of the gray mouse, and in the uterus of the white. Here the result was a black. So neither the ovum nor the uterus determines the color, while it's the nucleus. So this indicates the black color was coded by information inherited with the nucleus of the embryonic cell of the black, not the cytoplasm and the cell membrane of the ovum, and nor from the nutrition obtained from the white mouse. So deduce here, we can deduce that the genetic information is located in the nucleus. So dominant phenotype corresponds to an individual that can be homozygous or heterozygous, and here in this case, in the case in the book, this is ambiguous, so we have to watch for. If the descendants always have the same phenotype as of the parents, so the line is true breeding line, as we said. And after we get white mouse whose genotype is WW, so they should inherit W from each of their parents. They should be GW. Document 2, transmission of allelic genes. The transmission of the allelic genes is 44. We have two mouse. We have gray and we have white. They are crossed. Gray and white, they are crossed to GG, and white WW. So they are pure. These are pure. So only one type of gametes, which is G, and here only one type of gamete, which is W. So here in the first generation we have gray, they are hybrid, meaning that they are heterozygous. Heterozygous having genotype GW. So then we cross F1 with F1. Crossing F1 with F1, what do we call it? It's called self cross. Crossing F1 with itself. Give in the second generation, and this table of cross, GG 25%, GW 25%, GW 25%, and WW 25%. Let's start from the beginning. We did an explanation, a brief explanation. Let's move to detailed explanation. Here we crossed a gray female mouse with a white male mouse. All the obtained, they are gray. So first of all, you have to know that the gray allele is dominant over the white allele, which is recessive. This is the first indication. Second, you have to know that the parents, they are pure. This is DG, WW. Third note, you have to know that this cross is called a monohybrid cross. Why? Pay attention to this note. The parents are pure and they differ in only one trait, which is the color of the skin or the color of the coat. So this is a monohybrid cross where the parents are pure and they differ in only one trait, which is the color of the coat. Okay? In the F1 generation, first generation, we get a heterozygous gray mouse, meaning that it's gray, but it's heterozygous, having genotype GG or GW. For sure it's not GG, because GG is going to be homozygous or pure, but it's heterozygous or hybrid, it should be GW. So they inherit G from the gray mass and W from the white mass to have genotype GW. Upon crossing GW, G will give 50% and W will give 50%. So here we have GG25, meaning that it's a gray, and we have GW, which is 25, meaning that it's also gray since the gray is dominant. Also gray GW25, so it's a gray, and WW, it's white. Here we have gray 25 plus 25, it's 50, plus 25, it's 75. So 75% they are gray, while 25% they are white. These are the results of the self-cross upon crossing F1 with F1. So as a conclusion, after crossing a female gray mass with a male white mass, we got a homogeneous F1 generation. So we got homozygous F1 generation whose individuals have 100% G gray coat, meaning that they have GW. So the character gray is dominant over the character white color, which is recessive. Also the parents, they are pure. So let G capital be the symbol of the allele coding for the gray coat. Let W small be the symbol of the allele coding for the white coat. In F2, we get mice with white coat whose genotype is WW. White is recessive, so they should inherit W allele from each of their parents, who should be heterozygous, having genotype GW, as we said before. Homozygous heterozygous is always for the genotype, while homogeneous heterogeneous is always for phenotype. Factorial analysis, you have to start with phenotype, genotype, gametes, table of cross, table of results. Here, first of all, GG and WW. Here, 100% G, 100% W, so we'll get 100% GW, so they are with gray coat. And then we move to F1 cross with F1, genotype with F1, GW cross with GW, 50% G, 50% W, so we have 25, 25, 25, 25, we have 75% gray, 25% white, so the results are verified. Let's move now to the test of the Drosophila. We have Drosophila, two Drosophila, they are crossed. Drosophila with dominant phenotype, long wings, they said the dominant, crossed with Drosophila with recessive phenotype, vestigial wing. This is called test cross. Whenever we cross it with a homozygous, recessive one, this is called test cross. So here, long wings, it's dominant, so maybe it's going to be, it's dominant, so it's L capital. So L, or it can be L, V. Vestigial is recessive, it should be V, V small, V small. And the descendants, we get 100% Drosophila having long wings. If it's the case, we have only one phenotype in the descendants. It can be LL or LV. If it's LV, maybe it's going to be LV and VV. So it's not the case. So LV is cancelled here. So only have the chance to be LL crossed with VV. So it's LV. From this cross, we can determine the dominant parents if they are dominant or if they are recessive. In case 2, dominant phenotype long, either LV or LL. And here they should be VV. Here in the vestigial wing, they should be VV. So they should inherit V from each of their parents. So they shouldn't inherit V from here. The second V should be inherited from the long wings Drosophila. So they should be LV. So here we have two different phenotypes. So the parents are, the dominant person, they are heterozygous, having two different gametes. From here in the tested cross, we can determine if the parent, they are pure or they are hybrid. Case 1. After crossing a Drosophila having long wings with a Drosophila having vestigial wings, all F1 they are with long wings. So here we can conclude that parents are pure and the long allele is dominant over the vestigial, which is recessive, already mentioned. Let L capital be the symbol of the allele coding for long wings, and let V small be the symbol of the allele coding for vestigial wings. Drosophila, they have genotype VV, so they should inherit it from each of their parents. So Drosophila with long wings having genotype LL, it is P. Same case 2. After crossing Drosophila having long wings with the Drosophila having vestigial wings, we get in F1 50% Drosophila with long wings and 50% with vestigial. The vestigial wing Drosophila having genotype VV, they should inherit VV allele from each parent. So long winged Drosophila should be heterozygous, having genotype LV. After crossing the parent having red flowers, let's move to the flowers. Flowers, in this case, this one. Crossing red with white. Crossing red with white, we get 100% pink. What's the meaning of this? So we got a homogeneous F1, all of them, they are pink, which is intermediate between red and white. So the parents are pure. A third phenotype appear here. So this phenotype is intermediate. The pink one is intermediate between the red and the white. So this is the case of incomplete dominance. Both of them, they are dominant. Here we have RR. Here we have WW. So the pink have RW. Upon crossing two F1 individuals, RW and RW, we have 25% red, 25% white, and 50% pink. See, factorial RR with WW, the first cross, we get 100% RW. Crossing RW with RW, sure you have to do the table of cross and the table of results. So we get in the descendants, we get 50% RW, 50% RW here, and we have 25% RR, 25% WW. So the results are verified. Let's move now to this. This is showing the crossing between black, the crossing between female and male mice. We have 3 crosses. First cross, black cross with black. All of them, they are black. This is a normal case. The second case and the third case. Let's first talk about the second case, and then we can move to other cases. In the second case, we have something that is somehow not normal. We have crossing yellow mice among each other. We got the descendants having black coat. So the black is recessive with respect to the yellow, which is dominant. So Y capital, the symbol of the allele coding for the yellow, and B small for the symbol of the allele coding for the black. Black coated having genotype BB. They should inherit each of these alleles from each parent. So the parent that have genotype that are yellow, they are heterozygous, having genotype YB. The parents gamete are formed in equal proportion. This leads to 75-25, but we get 2/3, meaning that about 66.6667, and 1/3, meaning that it's less than the 1/4. It's less than 25%. It's 16 point something. So this can be explained that the allele Y is lethal. It causes the death of its homozygous carrier. So whenever we have homozygous, whenever we have YY, this will lead to its death. So this is what I have explained before. So black coated mice having genotype BB, so they should inherit from each of their parents. So the parents are heterozygous, having genotype Y capital B. Parents gamete are formed in equal proportion. This leads to 75% yellow mice and 25% in the descendants. We get 2/3 or 66.6% yellow mice. So the percentage of yellow decreased. This can be explained by the fact that Y allele is lethal. It causes the death of its homozygous carrier. So 25% if normal case, 25% YY, 25% BB, 50% YB. So phenotype, 25% they die. So here we're working above 75. So excluding them, we get 2/3 or 66.66 they are with yellow coat, and 1/3 or 33.333% with a black coat. Okay, this is everything for this session. See you next week for the next session. Bye bye.